Meiosis. Meiosis is the process when a single cell divides twice to produce a total of 4 cells. These 4 cells are our sex cells or gametes, sperm for males and eggs for females. These 4 cells contain only half the parent cell's chromosomes, a diploid.
Meiosis is important because it is essential in sexual reproduction. Without it, people wouldn't probably be here right now. This is the only process where it produces gametes.
However, where exactly does it occur, you ask? It occurs in the reproductive tissue. In male animals, it occurs in the testes, ovaries for females, and anthers and pistils in plants.
There is a difference between meiosis and mitosis. In mitosis, it is a cycle. New cells can go through cell division. Also, cells produced in mitosis are genetically identical to the mother cell, which contains a diploid number of chromosomes, while meiosis only produces gametes.
The first step in meiosis 1 is interphase 1. It is where DNA in the cell is copied, resulting 2 identical full sets of chromosomes. Outside the nucleus sits 2 centrosomes. Each has a pair of centrioles, which are structures that are critical for the process of cell division.
The second step is prophase 1. It is the step where chromosomes pair up. Each chromosome is made up of 2 sister chromatids, which has genetic information. These chromosomes may exchange bits of DNA, a process called recombination or crossing over.
The third step is metaphase 1. It is where the chromosome pairs line up next to each other along the center of the cell, sitting in the opposite sides of the cell, centrioles, which is extended by the meiotic spindles. These meiotic spindle fibers attach to one chromosome of each pair.
The fourth step is anaphase 1. The pair of chromosomes is pulled apart by the meiotic spindle. It pulls one chromosome to each opposite sides.
The fifth step is telophase 1 and cytokinesis. The chromosomes finish what they started and completely pull themselves from opposite sides. A membrane forms around each set of chromosomes to create 2 new nuclei.
A single cell then pinches in the middle to form 2 new separate daughter cells. This step is called cytokinesis.
Now, starting meiosis 2, the first step is prophase 2. Now, there are 2 daughter cells with 23 chromosomes each. The centrioles duplicate and the meiotic spindle forms again.
The second step for meiosis 2 is metaphase 2. Again, both daughter cells' chromosomes line up in the equator of the cell. Centrioles are on the opposite side and meiotic spindle fibers are back.
The third step is anaphase 2. The sister chromatids are pulled in the opposite poles because of the meiotic spindle. The separated chromatids are now their own individual chromosomes.
The fourth step is telophase 2 and cytokinesis. The chromosomes successfully separate and form a membrane around each set of chromosomes in order to create 2 new nuclei.
The last step is not completed with a new round of cytokinesis. Once there are a total of 4 granddaughter cells, each will have half a set of chromosomes.
In males, these 4 cells are all sperm, and in females, one of the cells is an egg cell, while the other 3 are polar bodies or cells that do not develop into eggs and just dissolve.
That is it for the meiosis documentary.
Have a great day!
And thanks for watching.